Hello students, my name is Komal Valani and I am Botany faculty in Shri Chaitanya Institute. In this session, chapter at a glance, we will quickly revise the complete chapter of respiration in plant from NEET point of view. Now from this chapter, usually 1 to 3 questions are asked in NEET. Now most questions are statement based, conceptual and can be diagram based also as ETS, Glycolytic Pathway and Krebs Cycle are concerned from this chapter. Now let us first see how many and what questions have been asked in previous year NEET exams. Now you can see that in NEET 2025 only one question came, in NEET 2024 only 4 and in NEET 2023 4 questions came from this topic. If you look at the question that was asked in NEET 2025, if I read the question was the complex second of mitochondrial electron transport chain is also known as. Okay, so the answer is succinate dehydrogenase. Yes, it is the complex second of ETS and is also called as succinate Q reductase. You might be knowing the name of the other complexes of the ETS also. Now you have to be very careful, students. You have to remember the name of the complexes of ETS because as it was asked last year and it has been asked previous year exams also. So you should remember the name of the different complexes of the ETS. Okay, let me tell you the name of the other complexes of the ETS also. You might be knowing that also. Now the cytochrome C oxidase is the complex fourth of ETS that is also called as cytochrome AA3 complex. Then NADH dehydrogenase is the complex first of ETS. Then cytochrome BC1 complex is the complex third of ETS and complex second is being succinate dehydrogenase. That was the question. Complex fifth is the ATP synthase or FOF1 particle or so-called oxysomes. Okay, so you will be getting an idea what type of questions are generally asked from this chapter, like this question came. I repeat this point again, you have to focus and do remember the name of the different complexes of the ETS and is going to be very, very, very important. Okay, so let us have a look. The next NEET 2024 question was asked this: which of the following are correct about cellular respiration? Now, these were the options given, and if I read these options, if you see, most of the options were the direct line of the NCERT textbook. Look at the first one. Cellular respiration is the breaking of carbon-carbon bonds of complex organic molecules by oxidation, and this is what the definition of the respiration and is given as such in NCERT. Okay, then the entire, is this correct? Yes, we know that respiration is an oxidative process. It's a catabolic overall, although it's an amphibolic process, this is what NCERT has given, but if we consider respiration and overall, it's a catabolic process. Okay, it involves the oxidation of organic substrates such as carbohydrate, fats and proteins, organic acids, etc. So this is right. Then look at the second one, the entire cellular respiration takes place in mitochondria. No, you know that there are two types of respiration, students, aerobic and anaerobic respiration. When we talk about aerobic respiration, generally it's a multi-step process and the first step of the aerobic respiration is the glycolysis if the substrate is glucose, okay, not fat and not protein. Although glycolysis is common for anaerobic also, but when we talk about aerobic respiration, the first step is the glycolysis, and glycolysis generally occurs where in the cytoplasm and is the first step. Rest of most of the part of the aerobic respiration generally takes place in mitochondria. It's not the entire respiration occurs in mitochondria, the glycolysis takes place in the cytoplasm. Okay, so see the third option then, the fermentation takes place under anaerobic condition in germinating seeds. Yes, it is true. You know that germinating seed at the time of the germination, there is generally the scarcity of the oxygen and initially the respiration is an aerobic one. Look at the D option. The fate of pyruvate formed during glycolysis depends on the type of organism also. Yes, it is true. You know that once the pyruvic acid is being produced as an end product of the glycolysis, it depends upon where it goes and which compound is going to convert into. That depends upon the nature and the type of organism and the presence and the absence of oxygen. If you remember that in yeast the pyruvate get convert into alcohol, and that's what we call alcoholic fermentation, while in case of muscles like in our body, the pyruvic acid in absence of oxygen get convert into lactic acid. So depends upon the nature of organism and availability of oxygen. Okay, so look at the E. Water is formed during respiration as a result of oxygen accepting electrons and getting reduced. It is also true. You know that water is an end product of ETS. Yes, and it's the end product of the respiration also, one of the product of the respiration. And we call it as a metabolic water also. And you know how water is produced as an end product of ETS. Oxygen is a terminal hydrogen acceptor, and it accepts the electron and protons and get reduced into water molecule. Okay, so we have the option A, C, D, E correct. So this was the question. So now you're getting an idea what type of questions are generally asked from this topic in NEET exam. Okay, like this question came in 2024. Okay, so let us proceed to the other questions that have been asked in previous year NEET exam. Okay. Look at this question, match the column. Okay, so again, what I have told you that this is the point you are going to focus, that the name of different complexes of ETS, again the same question in form of match the column. Okay, so ETS complex first is NADH dehydrogenase, complex second is what we call succinate dehydrogenase, or what I told you, succinate Q reductase also. ETS complex third is what we call cytochrome BC1, and the fourth is cytochrome C oxidase. Okay, so now let us see the next question, again the match the column. Okay, and now this is going to be very important, another point that you have to need to focus on, that where you have to remember the site of locations of the different steps of respiration. Like glycolysis site is cytoplasm, it occurs in the cytoplasm of the cell. Like the transition reaction occurs in the matrix of mitochondria, then comes the Kreb cycle and again in mitochondrial matrix, and ETS generally occurs in the inner membrane of mitochondria in case of eukaryote. If the same in case of prokaryote we talk about, glycolysis most of the part of the respiration in case of bacteria, even in case of aerobic bacteria, occurs in the cytoplasm only. Okay, while the ETS generally in case of bacteria takes place in the plasma membrane. Okay, now this was the question. If you look, identify the step in tricarboxylic acid cycle which does not involve the oxidation of the substrate. So answer is third, that is succinyl coenzyme A to succinic acid. Okay, so you know that succinyl coenzyme A, conversion of succinyl coenzyme A into succinic acid is not in a step of oxidation. Now let me recall that we have the three sites of oxidation in Kreb cycle. Okay, the first step is being the conversion of isocitric acid into oxalosuccinic acid is what the site where you will see the NADH is formed. Then the next site or the next step of the oxidation is generally where alpha keto glutaric acid gets converted into succinyl coenzyme A. Then we have the succinic acid get converted into fumaric, and finally we have malic acid convert into oxaloacetic acid. So we have the four different sites of oxidation in one round of Kreb cycle. So the conversion of succinyl coenzyme A to succinic acid is not a site for oxidation. Basically what happens, it's a site for substrate level phosphorylation, where we have a formation of one GTP molecule, and one GTP, as you know, is an equivalent to one ATP also. Okay, students, this was the next question, came in NEET 2023. How many times decarboxylation occurs during each TCA cycle? So the same type of question they're asking. So you're getting an idea what type of the question comes from which subtopic of this chapter. Okay, so you know that how many times the TCA cycle or Kreb cycle operates, that is 2 times. You know that how 2 times the pyruvic acid get converted into 2 molecule of acetyl, and because of the 2 molecule of acetyl, generally Kreb cycle occurs twice, okay, if the substrate is glucose, not a fat. Now these are some assertion reason question which have been asked previously in 2023. This was the assertion reason came. Now assertion, ATP is used at 2 steps in glycolysis. The first step is used in converting glucose into glucose 6-phosphate, and also the ATP is used in the conversion of fructose 6-phosphate into fructose 1,6 biphosphate. Yes, I think the ATP is used at 2 steps, so it is a correct explanation also, and both assertion and reason true. Now if you know if you recall, the glycolysis generally occur in two phases. There are two phases in glycolysis and 10 steps, which NCERT has also mentioned. Okay, so what are the name of these two phases? One is called as an activation phase, where the glucose generally get converted into fructose 1,6 biphosphate, and the second one is called as a payoff phase. So the first phase of the glycolysis is an investment phase of the ATP, where at two sites ATP is used. The first step where the glucose gets converted into glucose 6-phosphate, and at the second step where the fructose 6-phosphate get converted into fructose 1,6 biphosphate. So it's the correct explanation here. Okay. Now, this was another question: which of the following combination is required for the chemiosmosis? Students, you know about chemiosmosis. The chemiosmotic hypothesis was actually proposed by Peter Michel and is very well given in both the chapters of photosynthesis and respiration to explain the how ATP is formed. Okay, so let us have a quick look. So, which of the following combination is required? You know that the chemiosmosis requires first of all a membrane. Across that membrane you are going to create a proton gradient, and that proton gradient is required for making of ATP. Okay, so proton pump, proton gradient, ATP synthesis. I will discuss this in short in the coming lecture or the coming, okay, in the next slides we will discuss about it. Okay, so this was another question against the match the column. As you can see, they have asked to match the enzymes with respect to their the process they are involved in. Oxidative decarboxylation, glycolysis, oxidative phosphorylation, tricarboxylic acid. That means you have to remember now another thing from this chapter, what they are going to ask, the name of the enzymes also. So that not all enzymes are here important. Those, the name of enzymes that are given in NCERT are going to be very, very important. So you have to remember, you have to by heart the names of the enzymes also, okay, with respect to the type of reaction they catalyze. Okay, now this is the concept map of cell respiration. We are going to, I'm coming to the discussion of some of the important lines from NCERT textbook of this chapter. Before we go into that, let us first discuss about the basic concept of the cellular respiration, which I have already discussed while discussing these questions previously. So you just have a look on the concept map of the cell respiration here. It's a very easy form and you can summarize, okay, it's a simplified way what is happening in the process of the respiration. Okay, so if you look at this map very closely, the cell, you know that cellular respiration, or we have two type of respiration. Basically it is aerobic and anaerobic. If you look at aerobic respiration that occurs in presence of oxygen, generally it occurs in more than one step. The first step of the aerobic respiration is the glycolysis. No, glycolysis is the oxidation of glucose into pyruvic acid. So what happens in glycolysis and where it takes place? If you look at the location of the glycolysis, it occurs in the cytoplasm. What happens in glycolysis? One molecule of glucose get oxidized into two molecule of pyruvic acid, where you will have the net gain of the 2 ATP, the 2 NADH generally being formed as a reducing equivalents, and the end product being 2 molecules of pyruvic acid. Pyruvic acid is a third carbon atom, and students do remember, you have to remember the number of the carbon atom in that compound also. Suppose glucose is a 6 carbon compound, you must remember that pyruvic acid is a 3 carbon compound, and so as the other intermediate product being formed in the process of respiration. Got it? Okay. So this pyruvic acid further get transported into mitochondria from cytoplasm to participate in the next step of aerobic respiration, and that is the transition reaction. Okay. If you look at the transition reaction, where your pyruvic acid first converts into acetyl coenzyme A, and this acetyl coenzyme A then enters into the third step of the respiration, which is the citric acid cycle or so-called Krebs cycle. Now this transition reaction, that is the conversion of pyruvic acid into acetyl coenzyme A, generally occurs in the matrix of mitochondria in eukaryote. Now the Krebs cycle occurs in again matrix. Krebs cycle or citric acid cycle or TCA cycle, tricarboxylic acid cycle, generally these are the products of the citric acid cycle. So there are two decarboxylation reaction in citric acid cycle, so you are going to get 2 CO2. Here also in transition, where pyruvic acid get converted into acetyl, we have the loss of carbon in form of CO2. Generally glycolysis doesn't have any carboxylation, you have to remember that. Okay, so the other substrate level phosphorylation, you will get 1 GTP, 3 NADH and 1 FADH2 being formed, okay, in citric acid cycle. Okay, so if you look at now, these NADH and FADH2, they act as an electron carrier, and then they participate in electron transport chain that occurs in the inner membrane of mitochondria, and this is how the ATP is formed. And that process of formation of ATP, okay, via ETS is what we call oxidative phosphorylation, and the mechanism of the synthesis of ATP using energy from electron transport is what we call chemiosmosis. Okay, so this is what you are going to get from 1 molecule of glucose. You get a net of 36 or 38 ATP. Now sometimes you should not get confused that whether if it is asked, if it is come that from 1 molecule of glucose how many ATP are generally produced. As given in the NCERT, we have 38 ATP, sometimes 36 also is the net gain. Why so? Because you know that you are aware of the two type of the shuttle also, although not given in NCERT. If it is a glycerol 3-phosphate shuttle, then the net gain is of the 36 ATP. If it is a malate aspartate shuttle, then it is of 38 ATP. Got it? So just go through, have a look on this concept map of cellular respiration. Got it, students? So let us now start with some of the important lines of the NCERT textbook after this. So I'm projecting here the page-wise the NCERT and page-wise NCERT respiration in plants. Okay, and I have highlighted some important points from that chapter. So let us discuss. Okay. This is what the whole chapter is about and is what given in NCERT textbook, as you are all aware of. Okay, so this point what I have highlighted, we must remember that in green plants too, not all cells, tissues and organs photosynthesize. Only cells containing chloroplasts that are most often located in superficial layer carry out photosynthesis. Hence, even in green plant, all other organ tissues need food for oxidation. Now, what does it mean? Actually, you know that not all parts of the plant are photosynthetic. Okay, if you talk about the leaf and the stem, that are the aerial parts of the plant, are capable of photosynthesis, and that too only the leaf. Okay, in some cases the stem also. Root is being done underground and it is a non-green part of the plant. So what this line says that generally respiration occurs in all type of the part of the plant body. It's not like photosynthesis that it should takes place only in some part. Generally respiration occur in green part also, like stem and leaf, and it can occur in the non-green part of the plant body also, because every cell needs energy. Okay, every cell needs the food. Okay, so this is what line it means, that whatever the food is synthesized in the leaf or the stem, as a result of photosynthesis will get translocated, okay, with the help of the phloem to the non-green other part of the plant also, and they utilize that food and they oxidize it to get an energy. So respiration occurs both in green as well as the non-green part of the plant, while photosynthesis occur only in the green part of the plant. This is what this line means. Okay, now the next. Now if you look at, this is what the line which I have highlighted. This is a definition of cellular respiration, which you're all aware of what cellular respiration is. So if I tell you in the other words that overall respiration is basically you can say an amphibolic process, an oxidative process. As per amphibolic, as per the NCERT, that is both catabolic and anabolic, but overall it's a catabolic as it is a breakdown of the organic compound. Okay, and exothermic process just because of the loss of energy in form of heat. So heat is also liberated, so it's an exothermic process basically. Okay, then if you look at this, what happens in the respiration, as I've told you, it's a catabolic process, as there is a breakdown, as there is an oxidation of organic molecules here. Now the organic molecules that serve as a substrate, or you can say as a fuel, which burn and get oxidized in the process of respiration, we call them respiratory substrates. Can you tell me? Yes, let me tell you, these respiratory substrate that are being used as a source of energy in the process of respiration are carbohydrates, fats, there comes proteins, and then organic acid. You have to remember the order also. You know that glucose is an instant source of energy, so it is first carbohydrate get oxidized, and after the reserves of the carbohydrate generally get depleted, then cell oxidizes the fat, okay, to get the energy. and after fat during emergency, as you know, then the protein also serve as a respiratory substrate. So respiratory substrates are basically carbohydrate, fats, proteins, and organic acids also. Okay. Now remember that the fats have got more calorific value as compared to the carbohydrate, but they are very difficult to oxidize as they're oxygen poor compounds. Okay. Now this line, if you look at, I am reading this line for you. During oxidation within a cell, all energy contained in respiratory substrate is not released free into the cell. Yes, it is true. You know that we have to break the compound. If you want to oxidize and to get energy, complete energy from a respiratory substrate, you have to break it in small steps instead of just burning it in a single step. Yes, this is how you can compare here respiration with combustion. Now combustion is somewhat, it's a one-step process, and all energy of that hydrocarbon, okay, or a fuel generally is lost in one step in form of heat. Okay, but if cell needs and convert energy in needs an ATP and needs an energy, it has to break, it has to oxidize that compound in a small steps. That's why respiration is not a one-step process like combustion. In fact, it's a multi-step process. Okay, and I've already mentioned that aerobic respiration occurs generally in the 4 steps. The first step being glycolysis, which is common for anaerobic also. Let me repeat that same point again. The second step is transition reaction. Then here we have Krebs cycle, and then comes your ETS or electron transport chain. Okay. Now you know that, you should know that ATP is a chemical form of energy because cell use ATP only. Only the form of energy that cell utilize and use it for its function is the ATP only. So that's why the cell use food, because food is actually what? Food energy. Now it cannot directly use glucose as a source of energy for its function. It has to break the glucose and it has to convert it first into chemical form, and that is what we call ATP, which is an energy currency or the coin of the cell. Okay. And remember that, students, 1 ATP is equivalent to 7.3 kilocalorie per mole of energy. Okay. So do plants breathe? Yes, they plant breathe. This is what the line, what does it mean? Plant doesn't have the respiratory system like us animals have. They don't have the lungs. In fact, they have got an independent, each and every part of the plant body has got its own system of exchange of gases. Like leaf, we have stomata. Like in case of the bark of tree, we have lenticels present. Lenticels can also be present in the roots, as in case of pneumatophore. Okay, so this is what the difference between animal and the plant. Plant doesn't have any one point for the gaseous exchange. Each and every part of the plant has got its own system of exchange of gases. Okay, so this is about the next page. Okay, the key is to oxidize the glucose not in one step, which I have already discussed. Why respiration cannot occur in one step, why it should occur in multiple steps, and you know the reason why. Okay, so here let us start with the first step of the respiration, and that is glycolysis, which I told you. Glycolysis is a preliminary step or the first step, and is being common for anaerobic respiration also. But if the question comes, the glycolysis is an aerobic process or an anaerobic process, the answer that it's an anaerobic. By nature, the nature of glycolysis is anaerobic, although it's a different thing that it's the first step of the aerobic respiration also, but it doesn't require oxygen at that time to happen. Okay. If you look at the location of glycolysis, it is again cytoplasm, whether it is prokaryote or eukaryote. And what happens in glycolysis, you know, the one molecule of glucose get oxidized into 2 molecules of pyruvic acid. Along with that, we get a net gain of 2 ATP and the 2 NADH plus H plus. Do you know NADH is a reducing equivalent and it's an electron carrier. So it will then participate in the ETS, the last step of aerobic respiration, transfers the electron, and with that energy is going to produce ATP there. And what we call that processes as oxidative phosphorylation. Okay, and it's coming in the next slides. Okay. Okay, so this is a glycolytic pathway, students. Now you should remember that glycolysis occur in 2 phases, which I have mentioned. That then what are the name of these 2 phases? The first phase is called as activation phase, where glucose get converted into fructose 1,6 biphosphate. Do you know this glucose, fructose, and fructose 1,6 biphosphate are all esters. They are basically the esters. Okay, so up to here we call it as an activation phase because there is an investment of 2 ATP. Now in this case, you have to remember the name of the reactions also of this pathway. Let me tell you quickly. The name of the first step is called phosphorylation. Then second step is called isomerization. The third step is again phosphorylation. Then the fourth step is the cleavage or the splitting, where the fructose 1,6 biphosphate is split into 2,3 carbon sugars, that is DHAP and PGL. Okay, after that we have this step called oxidation. Then comes your substrate level phosphorylation, where you get the ATP what you have invested. Now now the system pay you for that. Okay, so here we have a production of 2 ATP, as we have 2 molecules here, and then we have the next 2 triose phosphate. This is intramolecular rearrangement or positional isomerism. This is dehydration, and this is again substrate level phosphorylation. Although glycolysis happens or occurs in 10 steps, NCERT has not given rest all 10 steps in this figure. This is a glycolytic pathway, and they haven't mentioned whether the step is reversible or irreversible. Let me tell you, out of 10 steps, there are 3 irreversible steps, first, third and 10th. Rest, most of the steps of the glycolysis are the reversible ones. Okay, and now you have to remember the reactions also, like oxidation happens for the ones. Oxidation is only one oxidation. There are 2 substrate level phosphorylation here. At least you should remember that there are 2 substrate level phosphorylation and 1 oxidation in glycolysis. What is substrate level phosphorylation, or SLP in short? Substrate level means where an ATP is formed as a result of the phosphate transfer from the substrate, and that to a direct one. Okay. Now, the synthesis of the ATP through NADH later on in ETS will be called as oxidative phosphorylation. So, students, remember these two. The ATP is formed in the process of the respiration by 2 ways, either substrate level phosphorylation directly from the substrate, and also the oxidative phosphorylation, that is through ETS. Okay. Now, coming to the next is the fermentation. Now, you know that fermentation is basically an anaerobic respiration, and generally it occurs in case of microorganisms such as an yeast. And yeast, you know that the product being alcohol, so we call it as an alcohol fermentation. So just remember the steps of the fermentation. By taking an example of yeast, you should remember what happens. The pyruvic acid will get first convert into acetaldehyde in presence of pyruvate decarboxylase, and then acetaldehyde will get reduced, okay, by the use of glycolytic NADH plus H plus to get converted into alcohol. Okay, so this what happens in the fermentation, and the same thing happens in the muscles of our body, where but the product is not alcohol, it is lactic acid there. This was the question asked in the NEET, that the fate of the pyruvic acid is going to convert into depends upon the type of the organism. Okay. Now, what then would be the maximum concentration of alcohol in beverages? That you know that yeast can tolerate only 13% of the alcohol which it produces. After it, the alcohol become toxic for itself. So, how? The question is the concentration of alcohol in beverages that are naturally fermented. You know that there are some liquors, you know that there are some distilled liquors like whiskey, brandy. These liquors have got a very high concentration of alcohol as compared to the non-distilled such as beer. Okay, so how? If 13% is toxic of more than 30%, the yeast cannot produce, then how 40 to 45% of alcohol is achieved in these liquors? So the answer is distillation process. Okay, this involves some of the concept of the distillation process of the chemistry also. Okay. Now, next is what we let us start with aerobic respiration after fermentation briefly. You already know many things about it. I have already discussed glycolysis. The second step is the transition reaction. What happens in transition? Quickly have a look. Pyruvic acid get converted into acetyl coenzyme A in presence of the name of enzyme is going to be very important here, as NCERT has given. So what I am telling you again, you have to in this chapter very careful about the names of enzymes which have been mentioned. Okay, so here the name is pyruvate dehydrogenase enzyme. The cofactor is magnesium. This can also be asked, where the pyruvic acid get converted into 2 carbon acetyl coenzyme A, and we have a 1 NADH also. Okay, actually there are 2 molecules of pyruvic acid being formed in glycolysis. So here we will get 2 acetyl and 2 NADH, and this reaction also gives you around 6 ATP. So 8 ATP in glycolysis, that is 6 ATP from 2 NADH, then net gain of 2, 8, and now here 6, total 14 ATP. being. This reaction generally occurs in the matrix of mitochondria, and we call it as a transition or a link reaction, or it is a oxidative decarboxylation reaction. As you can see, there is a loss of one carbon in form of CO2, and there is an oxidation also taking place. And student, remember this thing: wherever there is an oxidation in any step of the respiration, the name of the enzyme is always dehydrogenase there, and in most of the cases the acceptor of the electron or the proton is NAD. NAD is a coenzyme and it's a very, very good electron carrier. The full form of NAD is nicotinamide adenine dinucleotide, you might be knowing. Okay, so quickly, let me shift to the next page of NCERT. This is what the Krebs cycle is given. The Krebs cycle generally occurs in the matrix of mitochondria, but you are going to remember some of the most important products of the Krebs cycle. Let me tell you. The first product is a 6-carbon citric acid. This is the first 6-carbon product of the citric acid. This is how they will ask the questions. Okay, you have to remember like this, because we have the other 6-carbon products are also formed. So if they ask to the first 6-carbon product of Krebs cycle is citric acid, the first and the last 5-carbon product of the Krebs cycle is alpha ketoglutaric acid. The first 4-carbon is being succinyl coenzyme, which they have not shown here, and the last 4-carbon product is the oxaloacetic acid. Now you have to be very, very particular here where how many NADH are formed, at how many places the NADH plus H plus being formed in the Krebs cycle. At one side we have a production of FADH2 also. FAD is also a coenzyme like NAD, and the full form of FAD is flavin adenine dinucleotide. Okay, there is one substrate level phosphorylation where you can see one GTP is formed. This GTP get converted into one ATP. Okay, it's an energy equivalent, and here it get converted into ATP. Okay, so this is what the whole scenario. But let me tell you one more point. The question came previously in NEET exam that the Krebs cycle is an amphibolic cycle, although we say for the entire respiration, but Krebs cycle is an amphibolic because it is not only catabolic, it is anabolic also. Why? Because the intermediate products of the Krebs cycle are the precursor for the synthesis of the other compound also. And the same thing applied for the whole process of respiration, not only just for Krebs cycle. Okay, then we have the next and the last step of the respiration, and that is called what we call it as ETS or Electron Transport System. This is the last step of aerobic respiration, students. This is also known as terminal oxidation. You might be knowing about it. It is also known as respiratory chain or ETC, electron transport chain. It occurs basically in the inner membrane of mitochondria in eukaryote, and in prokaryote it occurs in the plasma membrane and perhaps the mesosomes also. What is ETS? ETS is the basic ETS that you are going to remember. You already know, okay, no need to repeat it. ETS is a transfer of electron, and with the energy of the transfer of electron, you are going to convert it into the ATP or the chemical form of energy. This is only the way to get an ATP, because you know that the law of conservation of energy says that the energy is neither created nor destroyed, but it is transformed from one form to another form. Okay, so one of the end product of ETS is water, and you are going to get the ATP from the NADH. NADH and FADH2 that have been formed previously in the other previous steps of the respiration. Now they will enter in the ETS and transfers their electron, donate their electron. Then you have to remember the name of the carrier and the name of the complexes of the ETS, which I have told you while discussing the question came in 2025. I repeat the name of those complexes that are involved in the process of ETS. Complex first is called NADH dehydrogenase. Complex second is called succinate dehydrogenase, which is also called as succinate Q reductase, and is the enzyme of the Krebs cycle also by the name of succinate dehydrogenase, and is located in the inner membrane. There comes complex third, which is cytochrome BC1. Then comes your complex fourth, cytochrome AA3 or cytochrome C oxidase, and of course we have complex fifth called ATP synthase or the FO-F1 particles. Okay, which is going to form an ATP. After this, now the question comes, there is always a confusion because in old NCERT they have given about 2 proton and now they have changed it to 4 protons. So what should we write? If the question comes, the number of the proton that diffuse from the outer chamber of mitochondria into the matrix at the time of the synthesis of ATP. So the answer is 4, as you can see in the figure. Now let me explain you. This is what an FO-F1 particle or ATP synthase that consists of 2 parts. this is the head part F1, this is the base which is called FO, and there is a stalk with which they are connected. This FO is being embedded and it's a proton channel. The head is projected. When the 4 protons used to diffuse from this channel, this F1 part become active to catalyze the synthesis of the ATP. So diffusion of 4 proton at a time will lead to the synthesis of 1 ATP. 1 NADH is equivalent to the 3 ATP and 1 FADH2 forms 2 ATP. Remember that. Okay, the calculation part here is going to be very, very important. I again repeat, student, this chapter, you have to remember the name of the enzymes, you have to remember the name of the reactions, you have to remember the calculation of the ATP wherever it is necessary, net gain, etc. So be aware of. Okay, now, so that no silly mistake is done from your side when the question comes. Okay, so this is a respiratory balance sheet which they have given. You know that they have calculated, we can calculate the ATP only when we will consider that respiration is a catabolic process. After considering, when we consider respiration as a catabolic process and is not, we are not considering that it's an anabolic also, then only we are going to calculate this much number of ATP is going to be formed. Otherwise, in nature, in cell, respiration is, it occurs, both catabolic breakdown and synthesis are going on simultaneously. So we cannot say the overall respiration is a catabolic. So we will calculate the number of ATP 36 or 38 only when we consider, and that is only theoretical possible. Otherwise, in nature, what happens? You know that the intermediate product being formed of the respiration, they act as a precursor for the synthesis of the other compound also. So the anabolic process and catabolic process, they go on simultaneously. Okay. Okay, students, so this is what they have mentioned, everything which I have told you about why the respiration or a Kreb cycle even is an amphibolic pathway. Let me give you an example to prove how a Kreb cycle is an amphibolic and how a respiration is also an amphibolic, that it is both catabolic and anabolic, by taking few examples here. If you look at the fate of pyruvic acid, although pyruvic acid is formed as an end product of the oxidation of the glucose and glycolysis, do you know pyruvic acid, we can use it to convert into acetyl coenzyme one way, or we can convert it into the other products also, like lactic acid in muscles, and we can convert it into alcohol, or we can convert it into an amino acid also. Okay, so pyruvic acid is a precursor for the synthesis of the other compound. Okay, now we have the fate of acetyl coenzyme. Do you know that acetyl coenzyme, apart from being entering into Krebs cycle, what are the other uses of it? Acetyl coenzyme is the precursor for the synthesis of fatty acids, okay, synthesis of the cholesterol, and is also required for the synthesis of the carotenoid also, okay, and also for you can say the cholesterol fatty compounds, okay, and ketone bodies, of course. Okay, so alpha ketoglutaric acid as the 5 carbon product of the Krebs cycle is the precursor for the synthesis of glutamic acid. Oxaloacetic acid, which is the last 4 carbon product of the Krebs cycle, is the precursor for the synthesis of aspartic acid. So this is how the products of the respiration are the precursor for the synthesis of the other compound as well. Okay, so just go through this figure. This is going to be very, very important, and this figures gives you a clear picture that how a respiration is an amphibolic pathway. Okay, now the linker molecule, as you can see the diagram very carefully, whatever the respiratory substrate as, when they get break, they all going to meet at a one common point, and that is acetyl coenzyme. So the question came previously that acetyl coenzyme is what we call the linker molecule of the respiration. Okay, even the fatty acid undergoes the oxidation, which we call beta oxidation. The product is acetyl, and acetyl is the product of the oxidation of the pyruvic acid also. So whether it is a breakdown oxidation of fat in respiration, whether it is of the carbohydrate, the acetyl coenzyme A is the only link that we can enter into Krebs cycle further of the aerobic respiration. Okay, then we have a RQ, or the respiratory quotient. Student, just remember the formula of RQ, the volume of CO2 evolved, volume of oxygen consumed, and do remember that RQ, it varies with the type of the substrate and depends upon the chemical nature of the substrate. And you have to remember the values of the RQ. Just remember that RQ is measured by an instrument which is known as Genong's respirometer. Okay, so just remember the values of RQ. For carbohydrate, it is equal to 1, for fat it is less than 1, and for protein is also less than 1. Okay, for fat it is 0.75. Now look at this last line, students. Why they have, let me explain you why they have written like this. What is important to recognize is that in living organism, respiratory substrates are often more than one. Pure proteins and fats are never used as. Now what does this line means? Why they have written that pure protein and pure fat are not directly used as a respiratory substrate? Why? Because protein is a macromolecule. It's a polymer of amino acids. So you cannot directly oxidize the protein. As it is a macromolecule, you have to first hydrolyze, not oxidize. That's why they have written. So the protein first undergo the hydrolysis in presence of a protease into amino acid, and amino acid then get oxidized. So it's an oxidation of amino acid, not the oxidation of protein directly. And so as fat also hydrolyzes first into fatty acid and glycerol, and then fatty acid undergoes the beta oxidation further in the respiration. So that's why they have written this line that why protein and fat in their pure form cannot be oxidized directly, because they get first hydrolyzed and then get oxidized. Okay. So just go through the summary, because sometimes the question comes from the summary of the chapter also. So just go through the summary, okay, and it is the all important points you will get in the summary. Okay, so this is the balance sheet, students, in front of you, and this gives you a clear picture about the count or the total number of the ATP formed in each and every step of the respiration. If you look at the stages of the respiration, glycolysis, now you have to take into consideration here that substrate level phosphorus, as we have the two way of the synthesis of the ATP. One is substrate level SLP, and through ETS it is oxidative. So you have to remember it separately. In glycolysis, we have two ATP being formed as a result of substrate level. Then through ETS, oxidative phosphorylation, that is NADH, we have 6 ATP, depends upon the shuttle, glycerol 3-phosphate, or it is a malate aspartate. Then this is how the total 6 of ATP being formed in glycolysis, including the ATP of SLP and ETS both. Then we have transition reaction, we have 2 NADH, so total of 6. Then Kreb cycle, you know that in 2 rounds of the Kreb cycle, we have 1 GTP, that is 2 ATP, 2 GTP formed, 2 FADH2, that is 4, and 6 NAD, that is 18, total of 34. So 34 or the 32 here. Okay, so this is how the total net gain of ATP 36 or 38, depending upon the type of the aerobic respiration. So this is very, very important from the exam point of view, NEET point of view, this calculation of ATP, and that too you have to remember it. SLP, substrate level phosphorylation number of ATP in each and every step, and also an oxidative phosphorylation step. Okay, so remember them separately. Okay, so this was a balance sheet. Now coming to quickly, let me tell you about high yield topics, and you have to prioritize and you have to give priority to these points respiration from respiration chapter is concerned. Look at the most important points which I have highlighted. First, glycolysis is the EMP pathway. Remember the key enzymes that are given in NCERT textbook of glycolysis, where it occurs, etc. And do remember link reaction, Krebs cycle. Then of course the oxidation, decarboxylation steps, the name of the steps, and of course the number of the ATP being formed, and the name of the enzymes also. Then we have electron transport, which I repeat, you have to give emphasis, you have to give more importance to the name of the complexes of the ETS. Okay, then remember the RQ values. Okay, remember these are some of the crucial points which I have already discussed and mentioned. So just have a look on this crucial points for the NEET exams, which have been displayed here. This is a rate limiting step of the glycolysis. The enzyme is PFK, phosphofructokinase, which is also called as a pacemaker of glycolysis. And just see rest of the other things, amphibolic pathway or the nature, and just go through that figure of ETS given in NCERT. And just remember, it's not 2 protons now, which NCERT has given the correction. You have to remember the 4 proton diffuse as at the time of the synthesis of 1 ATP. Okay, in chemiosmosis. So these are the, just go through the key differences and definitions that are related with this chapter, like respiration versus breathing, like respiration versus combustion. Then we have these 2 type of respiration, although not given in NCERT, but you know, depending upon the type of the substrate, we have 2 type of respiration. If it is the carbohydrate and fat, floating. If it is the protein, then protoplasmic. Then just remember the end product of the fermentation, number of the ATP being formed in fermentation. Okay, then we have direct NCERT based focus and last minute revision. Okay, so just go through and you can just see, okay, the name of the enzymes, calculation part, and common misconceptions that most of the students, they think that the respiration generally occurs only during night because the photosynthesis occurs during day. So it's not the case. You know that respiration is a 24-hour process, even if it occurs even in the daytime also. Okay, it can occur both in green and non-green parts of the plant body, although photosynthesis occurs only in the green part. Then we have not just at night, no oxygen in glycolysis, because glycolysis is an anaerobic process, and efficiency, you must calculate efficiency. From 1 molecule of glucose you get 36 to 38 ATP. That depends upon the type of the shuttle. Okay. Now, these are the top 5 mistakes the student usually make in this chapter. So you have to be very conscious, and you have to be very careful, okay, not to do these mistakes. These are some of the common mistakes that a student do in NEET exam, okay, when the question comes. Now, first of all, you just see the question carefully, read it carefully, what it is asking. If the question is regarding the calculation of the number of the ATP, whether it is asking the net ATP or the gross ATP, so just you have to be very particular. And then we have assuming respiration occurs only at night, miscalculating ATP from ETS, misunderstanding anaerobic respiration, ignoring the role of cytoplasm in mitochondria. And then these are the key tips to avoid the mistake. The study NCERT closely, focus on conceptual understanding, and practice PYQs, the previously asked questions, okay, from this chapter. Okay, so these are the discrepancy questions happened in NEET previous year question from this chapter. The total ATP calculation controversy was there in year 2002, 2005 and 2014. It came. The issue was the older NCERT books and test calculated the net gain of ATP from aerobic as 38. Newer NCERT updated understanding focus on net gain of 36 ATP. Although they have not given the shuttle, but if the question come what it is given in NCERT now in the new textbook, just follow that. Okay, then this was a discrepancy, the number whether it is 36 or 38. Then the modern questions now emphasize approximate or focus on specific steps like glycolysis or Kreb cycle rather than a single fixed number, or they explicitly ask for the net gain. Okay, then common intermediate of respiration, the question which biomolecule is common to respiration mediated breakdown of fats and carbon and protein. Now both acetyl and pyruvic acid are technically involved, but actually the right answer is acetyl coenzyme A, because acetyl coenzyme is a linker molecule of respiration. Okay, so this was a discrepancy. Incorrect statement for Krebs cycle, that was an issue in NEET 2017. Okay, confused whether the acetyl coenzyme joins with pyruvic acid or oxaloacetic acid, with the latter being correct. You know that it join with the oxaloacetic acid if it has to enter into NOT, but NCERT has not mentioned the transition reaction. Okay, what they have mentioned, but the thing is that the Krebs cycle starts generally with the condensation of acetyl coenzyme with the oxaloacetic acid. Got it? Okay, so these are the some of the questions which I have taken from NCERT exemplar of this topic, and I think you have gone through this question, so just have a look. Okay, ultimate electron acceptor of respiration in an aerobic organism is, you know, the answer is oxygen. Phosphorylation of glucose during glycolysis is catalyzed by, you know that wherever there is phosphorylation, remember student, whenever there is a phosphorylation reaction, the name of enzyme is always kinase. Wherever there is a oxidation reaction, the name of enzyme is always dehydrogenase. Wherever there is an isomerization, the name of the enzyme in most of the cases is isomerase or a mutase, as in case of respiration. Okay, pyruvic acid, the key product of glycolysis, can have many metabolic fates under aerobic condition. It forms, you know that in aerobic condition pyruvic acid get converted into acetyl coenzyme, that is called link reaction, but anaerobically pyruvic acid can convert into some other product also that depend upon the type of organism. Electron transport system is located in mitochondrial, you know the site. Okay, then which of the following exhibit the highest rate of respiration? Germinating seed, root tip, leaf bud, or growing shoot apex. Okay, then we have mitochondria are called powerhouses. You know that the growing shoot apex are generally made up of meristematic cells, and meristematic cells are you know that highly respiring cells, and they are always in the continuous state of division. Then we have mitochondria are called powerhouses of the cell. Which of the observations support this statement? You can just go through it. And the end product of oxidative phosphorylation is, of course, end product is ATP plus H2O. Then match the following, choose the correct option, you can just go through. So in the end, I just want to conclude and again repeat the same point which I have told you earlier, that you have to be very careful regarding the names of the enzyme in this chapter, the type of the reaction. You have to remember the sequence of the whole process, okay, whatever is given in the NCERT textbook. Okay, so all the best, student, and just have a you can say have a just attempt the questions very carefully. Okay, so just avoid doing silly mistakes. Thank you, all the best.